Bismillahirrahmanirrahim. I am Sidra Mehmood, and today is our lecture number 13 of nuclear analytical techniques, in which we will discuss the biological applications of radiotracers. First of all, we will discuss the key concepts that what we will actually discuss in this lecture. Our topic is biological applications of radiotracers. So we will discuss the subtopics that are autoradiography, radioimmunoassay, and DNA analysis that are related to the biological applications of the radiotracers. Now we will discuss the biological applications of radiotracers. Uh, Today, the largest number of applications of radiotracers is in biology and medicine. Because of large number of applications, it is beyond the scope of this work to review them in any detail. Instead, we shall focus on three specific applications, one that is very old, one that is middle-aged, and one that is adolescent in its scope, if not in its development. And we refer these very old, middle-aged, and adolescent applications as autoradiography, radioimmunoassay, and DNA analysis, respectively. Now, first of all, we will discuss about the autoradiography. The autoradiography is the oldest method of detecting radioactivity. In autoradiography, a radioactive sample is placed on a photographic emulsion. After a period of time, the film is developed and precise location of a radioactive matter in the sample is determined from the pattern of darkening on the film. Thus, autoradiography is usually used to locate radionuclides in a tissue section, gross sample, or chromatogram. Basically, in this technique, which is called autoradiography, the sample or the components of the sample are located. We can locate, or we can either uh, determine that which component is present through this technique. Now we discuss the in vitro and in vivo studies used in autoradiography. Uh, Before discussing the in vitro and in vivo studies of autoradiography, we will discuss what is vitro and in vivo and in vitro studies. In vitro studies are basically those studies that are carried out uh, outside the living body. We can say that we can take a living cell that is being taken out from a living body, and then we make many experiments on it, or we chemically add some different compounds on it to check the activity of those compounds on that living cell, but we are not working inside the living body. So this study is known as the in vitro study, while in vivo studies are those studies in which the components or the chemical constituents are injected inside the living body. So when any reaction occurs, or the activity which of which we have of the compounds which we have injected into the body can be observed only inside the body. So these studies in which we use different living animals like mice, cats, rabbits, dogs for our experimental purposes, we say those studies as in vivo studies. So in vitro autoradiography methods, the isolation of cellular components are involved, like we use DNA, RNA, proteins, or lipids, following followed by labeling with suitable radioisotopes. while in vivo autoradiography, radioisotopes are coupled with radioactive tracers, and those radioactive tracers are then administered orally or via injection, and the distribution of radiation is evaluated in thin tissue or whole body cryosections. In vivo autoradiography using laboratory animals is widely used in metabolic studies, disease monitoring, and new drug development experiments. Special techniques are used to get optimum spatial resolution in the image and to prevent artifacts. Those are known radiation induced darkening of the film. So optical or electron microscopes can be used to examine the images. The common radioisotopes that are being used in autoradiography include sulfur, hydrogen, carbon, iodine, or phosphorus. Sulfur in the form of sulfur 35, hydrogen 3, carbon 14, iodine 125, and phosphorus 32. These radioisotopes are commonly used in autoradiography, and they are used to determine the distribution of the radio-labeled molecules in tissues, cells, or cellular organelles, but also in the study of protein modifications and DNA or RNA sequencing. Now, another application of autoradiography is the positron-emission tomography. This also comes in the section of autoradiography. A diagnostic imaging method. It is a diagnostic imaging method in which we observe the biological processes inside the body. In a positron emission tomography scan, a radioisotope is coupled to a radioactive tracer and then introduced in the body. The radioactive decay from the radiotracer produces positron particles with the same mass as electrons but with opposite charge. When a positron and an electron collide, they annihilate each other and two gamma rays are produced, which are detected by a gamma camera. Basically, in this technique, the radioisotopes and a radioactive tracer are coupled with each other and then introduced into the body, and then the decay of the radioactive compounds or the radioactive decay from the radiotracer produces the positrons. These positrons are same as that of the electrons, carrying the same mass. However, only the one difference that is of positrons is the charge, and they carry the opposite charge than that of the electrons, and both opposite charged particles, positrons and electrons, when collide, they destroy each other, and in this way the two gamma rays are produced, and those gamma rays can be easily detected by using a gamma ray detector or a gamma camera, which is used or can be considered as a detector of gamma rays. Now, in biological applications of radioisotopes or radiotracers, we will discuss radioimmunoassay. Radioimmunoassay, we have discussed it in explained form in earlier. Now we will discuss its applications. Radioimmunoassay is a highly sensitive method of determining the amounts of hormones, drugs, vitamins, enzymes, viruses, tumor antigens, and serum proteins in biological samples. It is based on the immunological reaction of antibodies and antigens. One starts with either an antigen or antibody labeled with a radiotracer such as H3C14 or I125. So in it we can also use different biological samples like hormones, vitamins, enzymes, serums, or different biological secretions, etc. And the sample can be labeled with radiotracer or radioisotopes. Now in radioimmunoassay, we discuss the antigen-antibodies interactions. We have taken an example of antigens and antibodies. In a reaction mixture, we have antigens and antibodies present. For each antigen, we say that we have a antibody at which the binding site of the antibodies are used as an attachment for the antigens, while in our reaction mixture the antibodies are available in a limited amount. So the antigens will now have a competition, or they compete with each other to bind on the binding sites of the antibody. So if the reaction mixture has a radio labeled antigen present in it, the additional antigen added will displace the radio labeled antigen and will not allow it to bind to the antibodies. Like we have in our reaction mixture antibodies and antigens present, and if we have limited amount of antibodies, then it started a competition between the antigens to bind on the binding site of the antibodies. And along with other antigens, if we have a radio labeled antigen present in our reaction mixture, they also start binding with binding sites of the antibody. But if additional antigen is added, then they displace the radio labeled antigen and will not allow it to bind to the antibody. Now we will discuss the procedure, that how this antibody antigen and radio labeled antigen mixing occurs, and how the activity of the stable unlabeled antigen present in the sample can be measured. So first of all, we will mix the antibody and radio labeled antigen together and allow them to bind to each other. So for this we set them for some period of time, so they may bind together. After this we precipitate the complex from the solution, which means that after their binding we have precipitated the reaction mixture and precipitated the complex from the solution. So now we measure the activity of the supernatant. Supernatant is the topmost layer of the reaction mixture. We measure the activity which is being occurred in the supernatant layer, and this show that this activity is being shown by the unbound antigen, and we can say that this is the amount of the unbound antigen which is measured from the supernatant layer of the reaction mixture. Now we mix the same amount of antibody and radio labeled antigen together along with the unstable antigen sample. First of all, we have mixed the radiolabeled antigen and antibody together. When we have mixed the antibody and radio-labeled antigen together, we precipitated the mixture and measured the activity of the supernatant layer, which showed that the antigens that are not bounded to the binding sites of the antibodies are present in the supernatant. Now we mix the same amount of antibody and radio labeled antigen together along with the unknown stable antigen sample. Now we have added the unknown stable antigen sample into the reaction mixture along with same amount of antibody and radio labeled antigens. Again, the stable antigen will compete with the radio labeled antigen for binding sites on the antibody molecules. Both stable antigen and the radio-labeled antigen start competing with each other to bind on the binding site of the antibody molecules. Then the radio-labeled antigen will not be able to bind as it did before, as now a competition occur between that of stable antigen and the radio labeled antigen. So the activity of binding or the binding capacity of radio labeled antigen is not as same as it was before the addition of stable antigen. Again, we precipitate the complexes from the solution and measure the activity of the supernatant. Now, this activity is a measure of the amount of stable unlabeled antigen present in the sample. When we have mixed antibody and radio-labeled antigen together, that activity was the measure of the amount of unbound antigen. But now when along with radio labeled antigen, stable antigen is also added into the antibody mixture, then the activity is the measure of the unlabeled stable antigen present in the sample. So basically we have divided this procedure into two steps. In first step, we have used only antibody and radio labeled antigen together, in which we allowed them to mix and bind with each other, and then their activity is checked, and that activity is the activity of the amount of unbound antigens present. But when we have added the stable antigen along with radio labeled antigen into the antibody reaction mixture, then the activity is the measure of the amount of stable unlabeled labeled antigen present in the sample. So by constructing a calibration curve that shows the amount of radioactivity present in the supernatant after adding known amount of unlabeled antigen, one can determine the amount of antigen present in the sample. Thus, radioimmunoassay is a special form of isotope dilution analysis discussed earlier. So basically a calibration curve is being plotted to determine the amount of radioactivity present in the supernatant before and after the addition of known amounts of unlabeled antigens, and hence the amount of the antigen present in the sample can be determined by observing that calibration curve. And the most important advantage of this technique is that it is highly sensitive and it has high specificity. Like we can determine the desired component from our sample very precisely through this technique. And very small amounts can be determined through this technique, like some picogram quantities can be measured through radioimmunoassay. Now we discuss the DNA analysis being used in applications of radiotracers. As DNA analysis is a very popular technique in which different investigations or identifying of criminals, establishing paternity, and detecting of different genetic diseases is carried out. So it is most popular in news media uh, due to its purpose in identifying different criminals. So for DNA analysis, we basically use a technique which is known as DNA fingerprinting. And in DNA fingerprinted printing, we use different fragments or extracts of DNA, and DNA fragments or DNA sample is being collected from different samples of blood, skin, hair, semen, etc. As DNA is present in the every part of our cell. So that a DNA sample can be collected from any body part like hairs, skin, or any secretions like saliva, urine, or any other body secretions like sweat. We can take a DNA sample from those secretions. And DNA can also be collected from blood or semen samples. So the DNA that is collected from different sites can be cut into different pieces using enzymes that cut either side of a repeated sequence. So the restriction enzymes are used to cut the DNA into different fragments or pieces, and those restriction enzymes are restriction endonucleases or restrictase that cleave the DNA into fragments. So in this, basically we can use DNA fingerprinting technique in which DNA samples are being used, and those DNA samples can be collected from different parts of the body, and those collected samples are then cut into different pieces by using different restriction enzymes like restriction endonucleases and restrictase enzyme to cut either side of a repeated sequence. So the resultant DNA mixture of segments have the different sizes, and then those enzymes, the enzymes that have cut the DNA into different pieces, those pieces are of varying sizes. So then electrophoresis is used to sort the fragments by sizes. Electrophoresis is a motion of dispersed particles relative to a fluid under the influence of a spatially uniform electric field. It is a commonly used technique in a lab to separate charged molecules like DNA according to size. So in it basically the fragments are sorted by their sizes, and then those separated fragments are then allowed to react with the radio labeled gene probes. As first of all we have collected our DNA samples. On that we have to do different fragmenting techniques, like we cut this DNA into different fragments by using different enzymes, and then those fragments on the basis of their size are sorted out by using electrophoresis, and then at the end they are allowed to react with the radio labeled gene probes. As the gene probes contain radio-labeled specific fragments of DNA that bind only to those DNA segments containing a nucleotide sequence that is complementary to its own sequence that would be its matching strand in the DNA double helix. The original DNA fragments are then identified by the radio-labeled DNA that has reacted with them, usually by autoradiography. The physical pattern on the autoradiograph is the pattern of a DNA sequences and sizes. So in this way, the DNAs are matched with those of the particular DNA. Like in this way, we have determined the DNA of the suspected from that of the victim. And hence this DNA will allow to assist the criminal investigations as well as the identification of deceased person. Since the late 1990s, the Victorian Institute of Forensic Medicine VIFM has provided DNA identification services for deceased person to a Coroners Court of Victoria, utilizing nuclear DNA or mitochondrial DNA in its analysis. The nuclear DNA analysis in particular is a short tandem repeat analysis. We can say it as STR or short tandem repeat analysis, and it is routinely used at the VIFM to generate DNA profiles for deceased persons and reference samples to assist in familial matchings, and hence assist in the identification of the deceased person. However, the mitochondrial DNA, it is the inherited through the maternal line. So, unless a mutation has occurred, siblings and all maternal relatives share the same mitochondrial DNA sequence. Human mitochondrial DNA is comprised of a small circular genome that contains highly variable regions with sufficient variations to enable human identification. Its robustness has led to its use as a reliable forensic tool, and in recent times enabling the analysis of human remains from casualties of war or man disasters. So mitochondrial DNA is not as informative as DNA analysis, nuclear DNA analysis. However, mitochondrial DNA is very useful when sufficient quantities of nuclear DNA cannot be extracted for a case, be it from bone, teeth, or decaying tissue. So in recent years, mitochondrial DNA analysis has been used to assist in coronial case investigations ranging from missing persons where putative mother or child relationship have been discounted or filmed by mitochondrial DNA analysis to homicides. So this is all the applications of radio traces, the biological applications of radio traces. So at the end you have the assignment that you have to find the applications of any specific radio tracer in different fields. Thank you.